everything today, right, John?
Yep.
That was the only way of getting John on time.
All right. Well, good morning, everybody. It's a real pleasure to have Xuewu Sui present today. I just give you a little history about Xuewu. So Xuewu was a graduate student at Case Western during the time that Dr. Popczewski and I were there, and we met him for his PhD studies. Xuewu did a lot of really fantastic work on understanding carotenoid oxygenase biochemistry, and he published several seminal studies in that area. Did really well, had a fantastic PhD thesis, and moved on to postdoctoral work, and I think he got his top choice for a postdoc at Harvard. And he two bosses was not enough for him, so I think he chose a lab where he now has three bosses. So he's in the Walther, Farese, and Leo labs, and he moved there to kind of move back into his original, I think, love of lipid biosynthesis and studying the formation of lipid droplets and so on there. And he's done really fantastic in his postdoc. Just had a paper published in Nature, another paper published in Journal of Cell Biology, so we're really proud of him, and it's really a pleasure to have him here today to present his recent work. And so with that, I'll turn it over to John to do a formal introduction.
Hello. It's my pleasure to introduce Dr. Sui. This introduction might sound a little bit similar to Dr. Kaiser's. He obtained his PhD in pharmacology at Case Western Reserve University under our own very own Drs. Kaiser and Popczewski, studying the structural and biochemical mechanisms of carotenoid cleavage oxygenases. He is currently doing his postdoctoral fellowship at Harvard Medical School under the directions of Drs. Walther, Farese, and Liao, and he has been studying the structure and biochemical mechanisms of a triacylglycerol-synthesis enzyme, diacylglycerol-O-acyltransferase 1. He has also been studying the role of cypin, or BSCL2, in lipid droplet formation. Dr. Sui has received the American Heart Association Postdoctoral Fellowship Award and has been invited to speak at several conferences such as FACET Lipid Seminar, FACET Lipid Droplet Meeting, and the Structural Biology Seminar at University of Massachusetts. Dr. Sui has published numerous manuscripts in journals such as PNAS, Journal of Cell Biology, Nature Chemical Biology, JBC, and recently also Nature. So it is very exciting to have Dr. Sui speak for us today.
Hello, thanks Chris and Philip for the invitation. It's nice to see old friend in lab. So today I'd like to talk about our recent published work on synthesis of human triacylglycerol enzyme. So apparently I'm out of the ophthalmology, but this is a very fundamental process for fat biosynthesis. So I hope can give you some idea about this very important physiological process. Also, it's Friday morning. I'd like to take this a little bit relaxing. So if you have any questions, just stop me and let me know. Okay. So this project is collaboration between Farese Walther Lab and also Liao Lab at Harvard Medical School. As you all know, triacylglycerol, or TG, is an energy storage molecule. It has a glycerol backbone conjugated with 3 fatty acyl chain with a cell ester bond. The acyl chain energy condensed in this reduced hydrophobic chains can be oxidized to provide energy. And the length and the saturation of this acyl chain can be varied, but you can see this is a very extremely hydrophobic molecule, so it cannot exist freely in water. So when present in excess, they are stored in an organelle called lipid droplet, which has a neutral lipid core covered with phosphomonolayer lipids. Beside TG, it also contains other neutral lipids such as cholesterol esters and also retinol esters as well. So it's well known that fat is important for human health. Why? Because in humans, many organs are synthesized and secrete fat, including not only in adipose tissue but also heart, skeletal muscle, intestine, liver, and mammary epithelium. TG biosynthesis is quite important for those organs because they all synthesize and secrete TG. If this process has been disrupted, many human disorders occur. For example, not only obesity, but also if you cannot synthesize enough lipids, you got lipodystrophy. And overaccumulation of fat in muscles can cause diabetes. Okay? And in heart, cause cardiovascular dysfunction. And if the disorder happen in intestine, you got diarrhea and also protein loss enteropathy. Liver overaccumulation causing fatty liver and liver steatosis. If it's living uncontrolled, can develop to cancer. And also, TG is important for mammary gland development and lactation. So it is really central metabolic pathway. So biosynthesis of TG actually has been elucidated many years ago, also known as Kennedy pathway. So during this pathway, free fatty acid is activated by fatty acyl-CoA synthase to form fatty acyl-CoA, and fatty acyl chain can be seriously converted to the glycerol 3-phosphate by GPAT or glycerol phosphate isotransferase to form MAG, monoacylglycerol phosphate, and then further add acyl chain by acylglycerol phosphate isotransferase or AGPAT to form PA. Okay? And this phosphate group can be removed by phosphatidic phosphatase to form diacylglycerol or DAG. At this moment, DAG can either be used to form glycerol can be either used to form phospholipids or can be converted by DAG to triacylglycerol. And this reaction is catalyzed by diacylglycerol isotransferase or DGAT. And because this is a highly hydrophobic molecule, it is believed they are released back to the ER membrane, which can be used to form lipid droplet. Okay? And if we take a closer look into DGAT reactions, so the acyl-CoA or acyl donor is activated by DGAT and cleaved at this cell ester bond. And this acyl chain is transformed to the hydroxyl group in DAG to form ester bond and in order to form the TG molecule. And DAG is function as a acyl-acceptor. So actually in human or in mammals, there are two types of DGAT. They are distinct and belongs to different protein families. And DGAT1 is first cloned by Farese back to 1998. So it predicts to have several transmembrane domains at the ER. And the other one was first cloned from oleaginous yeast. And the mammalian one is also cloned by Farese like in the same year. And this guy almost looks like a rat. It has a small hairpin to locate at the ER and has a large C-terminal soluble domain, which is believed to be the active center. And both of the enzymes catalyze the same reaction, convert fatty acyl-CoA and DAG to the TG molecule. And because TGA biosynthesis is a central metabolic process, several inhibitors targeting two different enzymes has been developed actually. And I think inhibitor against DGAT2 is being used to treat human metabolic disease in hospital. And inhibitor against DGAT1 has entered phase 3 clinical trials. And both enzymes indeed catalyze almost all TG in the cell. For example, this is a nail red stain of neutral lipids, and this is a cell from mouse. This is a wild type. You can see this red dot are the neutral lipid. However, if you knock out both DGAT enzyme, virtually there's no TG form. And also in the cell, you have this fat body or lipid droplet in the wild type. However, in knockout cells, it's almost gone. Okay. So the discovery of ACAT or DGAT is actually based on the sequence homologue from ACAT, which is acyl-CoA cholesterol transferase. It catalyzes very similar reactions. And the only difference is acyl-acceptor is cholesterol. And the final product is cholesterol esters. And DGAT is cloned based on the sequence homologue on ACAT by Farese. And this is the actual film from them. So they overexpress this enzyme, and compared to the ACAT overexpression of the wild type, they don't see too much cholesterol ester formed, but they increase the amount of TG. Okay, that's how they found DGAT enzyme, DGAT1 actually. And besides that, DGAT also is a promiscuous enzyme. It can recognize not only DAG, but also long-chain alcohol or retinol as acyl-acceptors to form waxes and also retinol esters, respectively. And today my talk will be focusing on DGAT1. And actually, DGAT1 belongs to a large enzyme family called membrane-bound O-acyltransferase, or MBOAT. So human has about 16 MBOAT enzyme. What they're doing is they recognize different acyl donor, for example, acyl-CoA, and they transfer the acyl chain from the donor to the acceptor molecule, okay, to generate this product. And based on the acyl-acceptor, they can be divided. Human mammals MBOAT can be divided into 3 different classes. So in these types, the neutral lipid is the final product, which contains not only DGAT but also ACAT, or you can call it SO1 or SO1 and SO2, the catalyze cholesterol ester formation. And some of the enzyme family, for example, H-hat or porcupine or goat, recognize protein as acyl-acceptor. Okay? And the remaining MBOAT recognize lysophospholipids as acyl-acceptors. And here we include another one from bacteria, and I'll show you why in a second. So some basic biochemistry about DGAT1 is after cloning of DGAT1 chain and the other orders from DGAT1 tends to form a larger oligomer. So here they use protein cross-linker. If you increase the amount of this cross-linker, you see not only dimer, but also tetramer and higher oligomer formed. And that's why the DGAT1 may tend to form a larger oligomer. And another important biochemistry for DGAT1 is their activity can be drastically increased by anionic phospholipids. And this is about this year. This is I think this is a brassica DGAT1 microsome, just a crude lysate extraction. You can say PA almost increased activity by more than 2-fold. Okay? And later on, another group, by using purified enzyme, they can see this is the kinetic curve. You can see when you add a PA in the solution, the maximum velocity can increase at more than 3 times. Why this is important? Because TG is an important biofuel product. If we can find out what's going on there, this is very important to increase oil production in agriculture and biofuel industry. So as a lab focusing on lipid metabolism and lipid droplet formation, we are really interested into how DGAT1 convert the soluble or semi-soluble substrate to the hydrophobic TG product, which is finally end up a lipid droplet. We really want to understand the underlying biochemistry and the cell biology mechanism. So to answer this, we choose DGAT1 and use structural biology approach, trying to delineate the underlying catalytic mechanism. So here we express human DGAT1 in hex cells and purify them in detergent first. I think this is a digitonin. So we got 2 peaks, larger peak and smaller peak, and also peak 3. So if you look at SDS page, peak 1 and peak 2, they all contain DGATs, and peak 3 is the MBP tag we use for purification. Okay? And if you look at the particle distribution of the major peak, which is here, they all look like this round shape overall disk, and here is the 2D average. So not only the larger particles, you can also see some smaller stuff, which we don't really know what are they, but the purity is quite good. So I guess there could be some minor contaminant protein. And from the 2D average, you can only see this roundish shape of the average, which is not ideal, but it's also not that bad. So we decided to take this prep and using cryo-EM to try and determine the structures. And this is one micrograph of the EM map. So you can see we even increase the concentration to 18 mg per ml to allow them goes to the battery's ice. And to my eyes, this is quite good. Okay, you got a lot of particles in one micrograph, and the contrast is pretty good. And by looking at this image, you can get some taste how this particle looks like. So they appear like roundish particles, and this is some side view as well. So pretty much looks like a disk. Okay, and we do collect almost 9,000 of images on this sample. And however, if you look at the first round of 2D average, okay, and we divide it into 6 different classes. So you see this overall shape, and this is a cut-through view. The problem is if you look into the membranes, okay, the signal is quite weak, and here in these 3 classes, the signal is almost gone, okay, which means the protein in this detergent are fairly flexible, and this is almost a very bad sign for this prep. And not surprisingly, when we further process this data, we got nothing. So this is pretty frustrating because if you from the micrograph, they look pretty good, and here you got nothing. So then we have to try something else. So I guess many of you heard about nanodisk, so which you can reconstitute your protein of interest into native lipid bilayer, which are circulated by memory scaffold protein. Okay? And that's what we tried next. I tried different lipids, different scaffold protein to reconstitute DGAT1 into the nanodisk. And finally, I reconstituted DGAT1 in POPC, which are circulated by MSP2 and 2 based on the size of the 2D average shown by negative stain. And here is the major peak. Okay. And if you look at the negative stain EM, they all look similar like before. It's a roundish shape. And I think the homogeneity is a little bit improved. And actually, this took me almost a year to optimize this sample. But we did the same thing, collect a large sample on this, and we didn't resolve the structure at all. So this rod seems not reasonable. All right, so now detergent sample failed, and the nanobig sample, which we're hoping a lot, also failed. So we have to try something else. Okay, and finally, what we tried is amphipause. So we reconstitute two peaks you just saw from detergent sample and reconstitute them into amphipause. And similarly, we got two different species, the larger species and the smaller species. Okay? And if you look at the particles by negative stain, EM, so this is the first peak, like similar to what we saw before, this disc-like structures. So this is the top view, okay? And this is the side views, and this is the 2D averages. And if you look into the second peak, so similar overall disc-like structure, but the size is much smaller than the previous one. As you will see, and here is the 2D averages. Okay, so to my eyes, the homogeneity is much more improved than what we saw in either detergent and also nanodiscs. So because as you will see later, we found this is actually the dimer of DGAT1. So based on the previous publication and also the size of this disc, we think this could be the tetramer format of DGAT1. So to continue our structure study, we actually choose the second peak, which appears more homogeneous by negative stain and also higher yield. But before structure study, we really want to make sure the purified enzyme is enzymatically active. So this is what we did. So we mix the acyl-CoA, which is radio labeled at the acyl chain here, and also provide that, mix them together to generate TG and extract them by organic solvent and run them on TLC. Okay, so this is DGAT1 in microsome, which we didn't purify at all. You can see the final product of TG, and its activities can be specifically inhibited by DGAT1 inhibitors. And in digitonin and also in PMA, our protein are also active, which is good. Although the activity of DGAT1 in PMA or amphipause is slightly less than that in digitonin. So everything seems good. We got homogeneous particles, and the particles is also active. And finally, we also measure the tetramer versus dimer activities. This seems comparable. So as I said, we choose the dimer peak for our structure study, and we finally managed to put them in ice. So you can see this is particles. I highlighted few. They look like what you just say in negative stain EM. And from the 2D averages, you can see this overall disc-like shape. But what important thing is in the membrane, I think the side view, you can see the helical pitches in the membrane, which is a very promising sign for higher resolution. And this is how we process the data. We collect almost, I think, 8,000 images, got about over 2 million particles. And we divided them into two different subsets because the number is too high. It would be very hard for computational study. So the first group, as you can see here, not like what we saw before. For example, in the membrane here is almost empty, but what exciting us is in the membrane, for example, here in this subclass, you can see the alpha helices density in the membrane, which is quite indicate, okay, this data set should work. And similarly, in the other half of the particle subset, we also see that in the membrane, it shows up the alpha helical signals. So we further combine the particle in these two groups, and also here, combine them and do another round of classification, but here we use C2 symmetry. Okay? Here we didn't use, we implied no symmetry there. And this number one stand out because it contains the most particles and also with the highest estimate resolution by relying on software. So we keep the particle in this subclass and then do a refinement, get an initial map, which I think is around 5 angstrom, and we use that as a reference. And by taking the same particle subset, we did a non-alignment classification, and immediately we got two classes. Here you can see not only the alpha helical density, but also the side chain density also appeared on the alpha helices. And also another here also showed similar thing, because this group has about 4 angstrom resolution. We take this and refine the final structure after 3D refinement and also per particle CTF correction and beam tilt correction. We end up with 3 angstrom resolution final map. And this is the sharpened map. If you don't sharpen the map, you can see this is a TM. This TM region appeared, which indicate this should be the micelle signals. So you can see the protein itself is mostly embedded in the membrane. Okay, and this CTF curve, FSC curve, and the particle distribution. You can see the particle mostly appeared as the side view. Okay. This is a final local resolution. As you can see here, in the central part, we can even reach about 2.5 angstrom resolution. And overall, the resolution is at 3 angstrom. And outside part is a little bit low, which is reasonable. And if you look at the side chain density, I think this is not worse than like even 3 angstrom crystal structure. You can see most of the side chain in the membrane. Some of here TM1 to TM9, and this is some other regions. And most important thing is catalytic histamine. The side chain density is well resolved. Okay. I hope here you can feel a little bit about the power of cryo-EM. So this is the final map. As I told you, in the final map, we actually see 2 DGAT1 monomers, and they form this domain swapped intertwined area to form this dimer. And because this overall resolution is about 3 angstrom, which allows us to build the model very confidently. But we do have some missing regions, like for example, very N terminals and very C terminals and the middle part. So the full length of DGAT1 is 488, by the way. And this region probably has a high flexibility, which we don't resolve the map at all. If you look at the topology of DGAT1 monomer with respect to the ER membrane, you can see, as I said, protein itself is mostly embedded in the membrane. And it has total 9 transmembrane domain. Each of them are connected by either short helices or the loop region. Okay? And the dimer interface is mediated by the N-terminal soluble region at the cytosolic site, and also TM helices 1. So then the next question is, where is the reaction center located? It has been speculated for a long time where is the DGAT1 active center. Some people proposed it at the cytosolic site. Someone said it at the ER luminal site. So it turned out to be the active center in our structure is actually embedded in the membrane, which has this extremely conserved histamines, as well as a few hydrophilic side chain. For example, there's 2 glutamines and 1 asparaginase locating around surrounding this histamine. And also in human DGAT1 structure, the histamine seems to form a hydrogen bond with a nearby methionine as well. So to validate this region is critical for the catalysis. We mutate each of them to alanine and measure the mutant activities. You can see they all decrease DGAT1 activities, but 2 of them, histamine and asparagine, which is here and here, they are essential for the catalysis. So taken together, we show this region is a catalytic center and is embedded in the membrane, the ER membrane. Okay? And if you look into the dimer interface, actually, this is a pretty, sophisticated interactions. So this part here is mediated by the hydrophobic interactions, which is mediated by transmembrane 1 and also with the opposing subunit by mainly hydrophobic residues. And also we also see this region in the cytosolic side from this hydrogen bond network to further support the dimer formation. So you can see this interaction are quite elaborate, and that's why we believe this dimer could be the functional subunit in the cell. And what if you remove this region to force the protein from the monomer? So that's what we did. You can see here we start to see protein from the hydrophobic interactions at 85. So what we did was we removed either N-terminal 85 amino acid or total 90 amino acids. So this is what you see here is we fuse the protein with the GFP tag and run gel filtration on the cell lysate by monitor the GFP fluorescent. Okay, and here is the wild type. You can see if you remove the N-terminus at the cytosolic site, the expression level got drastically decreased. Okay, and here is the peak, and the free GFP is supposed to migrate at this shoulder position. So based on the size of the position of this peak, although we don't have solid data for this, but we believe if you only remove the N-terminal region at the cytosolic site, protein should still form a dimer, but it needs more validation. And here is the the Western blot. So if we measure the mutant activity compared to the wild type, you can see they are still active, but the activity is much more decreased, which means the dimer formation could be important for DGAT1 to exert activities. So then our next question is now we have a good understanding about DGAT1 overall structure, which has a 9 total transmembrane domain, and the reaction center is in the membrane. Okay, so now next question is how DGAT1 capture the substrate. And so the first thing we focus on is the fatty acyl-CoA substrate. So what we did next is we incubate DGAT1 with 1 millimolar oleoyl-CoA, which is the preferred substrate. And in the final cryo-EM map, you can see here, we see additional this strong density appear in the final EM map. So someone may wonder, what if you don't add the oleoyl-CoA substrate? And this is how it looks like. Only has a very weak density in the region, and both of them are contoured at the same level, which we believe this could be some phospholipid bind at this tunnel. And this density can be perfectly fit by an oleoyl-CoA molecule, which you can see here. The protein inserts the acyl chain, which is hydrophobic, into the protein, leaving the coenzyme head at the cytosolic site. Most importantly is this cell ester bond, which is supposed to be cleaved during the reaction, locates next to the conserved histone, which further confirm here is the reaction center. And if you look at the overall density, not only the acyl-CoA substrate, but also the side chain are also very well resolved. Okay, for example, this histone. Okay, and the thing is, the region at this part, like acyl chain, also phosphopentyl group of the substrate are well resolved, and if you increase the contour level, the density, which indicate this region is relatively flexible and may not contribute to the interaction too much. And here is the detailed acyl-CoA DGAT1 interactions. Here means the hydrophobic interactions. Here means hydrophilic interactions. So we selectively mutate most of those hydrophilic residues to alanine. As you can see, they all decrease the activities, but not that drastic. Okay, so to figure this out, we select these 4 hydrophobic residues deeply in the tunnel and mutate them to larger side chain residue, for example, tryptophan and phenylalanine. As you can see here, this valine to tryptophan, which is here, and also the stearin to tryptophan, which is here, almost killed enzyme, which validated our model. And we do measure the folding of each mutant in our study by these GFP signals, and those data suggest they all folded well. This indicates the loss of the activity is not simply due to protein misfolding upon mutation. Okay. So based on our structure, you can see the only distal end of the acyl chain is interacting with the protein. So this suggests, okay, if you use a shorter acyl chains, the activity is supposed to be decreased because they won't be coordinate very well to hydrolyze this cell ester bond, and that's what we saw. So this is what we see with oleoyl-CoA, but if you decrease acyl chain to 10 or 12 carbon, the activity is much more decreased, which is consistent with our model as well. Okay? And if we compare the structure with or without oleoyl-CoA substrate, what we saw is the protein itself, like a rock, we don't see too much conformational change, but in the active center, we do see the histone is flipped to this side upon acyl-CoA binding and form this hydrogen bond with very conserved glutamine. Although we don't know if this conformational change is critical for the catalysis because we don't have the other substrate, which is a DAC, but appears to be this is the only conformational change we saw in the structure. So this could be the functional important for the enzyme. Okay, and here's the other essential side chain asparagine. So now we got a good idea about DGAT1 structure and how DGAT1 capture fatty acyl-CoA. Then how about the other two things, the DAC substrate and also the TG product? So if we look at the DGAT1 structure again, so this is a cut-through view, and this is the reaction center, and this arrow indicates the binding site of the acyl-CoA. We do see the active center is opened to the membrane by this lateral gate. Okay? So this immediate caught our attention. Why? If you imagine the TG product, which is hydrophobic, is supposed to be released to the ER membrane. And also that substrate, which could also be partitioned into membrane, if you want to bind the DIG substrate and also release TG product to the membrane, this lateral gate can perfectly serve this function. To further confirm this idea, if you remember this phylogenetic trace, there's one outlier I put here. This is called DLTP from gram-positive bacteria. It is also an MBOAT enzyme. What it does is it transfers alanine group from another enzyme protein called DLTC. Here is not the donor is a protein instead of lipids. So it transfers alanine group through also the active center of histamine in the membrane to the LTA or lipoic acid at the extracellular site to this OH group. So this reaction is the product on the extracellular domain is critical for cell wall biosynthesis. So you can see, unlike DGAT1, the donor is from one side of the membrane, and the acceptor is from the other side. Okay, it's on both sides of the membrane. Seems DLTP don't need to open a hole in the protein. And if you look at the structural comparison of two enzymes, two MBOAT, you can see. So this is DGAT1. This is a lateral opening gate to the conserved histamine, and here is the acyl-CoA binding site. You can see they are aligned very well. This part here is for the DLTC binding. It's almost identical compared to DGAT1. However, the distinct difference is, unlike DGAT1 is opening to the membrane, here the lateral gate is blocked by the transmembrane 1 and transmembrane 2. Instead, DGAT1 uses transmembrane 1 and the N-terminal to form a dimer. However, DGAT1 DLTP is actually a monomer. So since during the evolution, DGAT1 utilized N-terminal to form a dimer and also open up a hole in the membrane. So this is the overall lay of the two structures. The RMSD for the CF is quite close, like 1.4 angstrom. They're highly conserved. So if you look at the cut-through view again of DGAT1 and the DLTP, like I said, the alanine donor or acyl-CoA binding tunnel is highly conserved. Both enzymes has a membrane-embedded active center. However, unlike DGAT1, here is blocked in the membrane, and here is a cartoon representation. So this further indicate this lateral gate could be functional important for DGAT1. And to further confirm this idea, actually, very interesting thing is in our either substrate-free or substrate with acyl-CoA, we do see in this lateral gate, which is here by this dashed red line, we do see additional large density, looks like lipids. So this is not belongs to protein, and we believe this could be a native bound lipid molecule that co-purified with DGAT1. Okay, and in the acyl-CoA structure, this density just pointing orthogonal to acyl-CoA substrate, and they locate very close. And this is how the density looks like in the apo structure. The conserved histamine is located right next to this linear density. Okay, and if we look into the structure around this region, you can see not only highly conserved histamine and also asparagine. Both of them are essential for the catalysis, and also many hydrophobic residues together construct this linear density, linear pocket, to accommodate this linear substrate, potential linear substrate. And we do show this region is important for the catalysis because, for example, if we mutate this leucine to tryptophan, which is here, tends to block this tunnel, the activity is like only about 10% left. And if this region is the binding site for that, which is linear, it also can explain why DGAT1 can also recognize other substrate, for example, fatty this is a fatty alcohol, to generate wax, but cannot recognize other lipids such as cholesterol, which is actually recognized by ACAT enzyme. Okay? So as I told you before, DGAT1 is cloned based on the structural homolog on ACAT, which is generate cholesterol ester instead of triacylglycerol. Actually, in the same issue of Nature, there are two papers published to report ACAT structure. And in this paper, they resolved the tetramer format of ACAT1, but here I only show you the dimer. So they say the acyl-CoA bind at the same equivalent site as that in DGAT1. Okay, this is pretty conserved. However, this also see this this lateral gate is also exists in ACAT. And most interesting thing is they show here is bound to a ACAT inhibitor, which is nivolumab. I think this drug is being used in treating cancers. So they also, by doing docking assay, they show this lateral gate is also the binding site for cholesterol, which is consistent with our hypothesis as well. So with all this, I think we are very comfortable to propose a potential mechanism which the acyl-CoA either dissolved in the cytosol or partitioned into the membrane depending on the acyl chain length, then bind to the DGAT1 active center through this hydrophobic tunnels facing to the cytosol. And that substrate that can be partitioned into membrane and bind to the reaction center through this membrane-embedded lateral gate. So once the donor and the acyl-acceptor bind in their position, with the help of this histamine, the acyl chain is transferred to the hydroxyl group in the substrate to form the final hydrophobic TG product, which is most likely released through the same gate back to the membrane, and the other product, free coenzyme A, is released back to the cytosol. So you can see in ACAT1 structure, the inhibitor is bound to this lateral gate. So if I let you to guess, if we want to generate DGAT1 inhibitor, which could be the targeting site, you can think not only here, but here could be another site for DGAT1 inhibition, right? And that's what we are doing now. Actually, there are several very potent DGAT1 inhibitor has been used, and developed, and they are all very potent with nanomolar inhibition affinities. So far, we are able to confirm one of them, which is very first generation of DGAT1 inhibitor with 50 nanomolar inhibition potency. Actually, in our final structure, which is here, the density fits pretty well to the drugs, and this part is actually the acyl-CoA binding site, which is here. So this is something we're still in progress. We're trying to try different inhibitors to elucidate the inhibition mechanism on DGAT1, but at least we can see these drugs block the DGAT1 activities by occupy the acyl-CoA binding tunnel facing to the cytosol. So to summarize what I showed you here, by showing cryo-EM structure of DGAT1, we can see how DGAT1 captured the substrate and how DGAT1 released the TG product. So actually this releasing of TG product is very important for our research in lipid droplet formation, because we believe this TG products released to the ER membrane, and once TG released in the membrane reach a certain concentration, they form lens, and the following step will be the lipid droplet formation. So today I don't have time to talk about that, but on DGAT1 we still have a lot of unknown remained. So I think maybe Philip also interested in this, what the catalytic role of hastening during this reaction, because as you know, hastening can either be proton acceptor or proton donor. So during this catalysis, we really don't have no idea what the role of hastening and also the other essential side chain, which is asparagine, doing during the catalysis. We are working on that. And how DGAT1 recognize other acyl-acceptors, such as, I didn't mention that, that fatty alcohol and also how DGAT1 can be, stimulated by negative charge phospholipids, and also, I didn't talk too much here, also regulate by organization. And also, as I said, DGAT1 inhibition mechanism. So, finally, I really like to thank my postdoc mentor, Bob Toby, and also Walther. They give me a lot of support and allow me to freely explore on different things. I really appreciate that. And also, people help me with this project. And the EM facility at Harvard, UMass, and also HHMI. And of course, find out funding source for this project. And lastly, because I found the same lab, I really want to take a second to, bring me my old memory to the lab in Chris' lab. So this is me and Philip at the APS. And also here is the moment near my graduation. We finally resolved a structure we are, working on for almost 5 years. And this with labmate at the retreat. And this is one way, sending in to other positions back at the case. And with all that, I'd like to take any questions, and thank you for your attention.
Bravo, Xuewu, thank you very much for the seminar, and we're very proud of the progress you have made, and really you left a lot of friends behind. Let me just ask you quickly 2 questions, and then open up to a question of everybody. So do we know any human mutation that inactivate this enzyme? Is this enzyme lack of the DGAT lethal in humans? Most likely, if there is nothing like that. And are you pursuing also retinol binding site for this enzyme?
Okay, first question, any human mutation on DGAT1? Answer is yes. So there are a few mutations, and we also recently published a paper to, characterize one mutation. So mutation in human DGAT1 causing very severe diarrhea. So in children with DGAT1 mutation, they, go to bathroom a lot. So those patients normally cannot survive, so they die early age. And the second question is retinol. I think the binding site for retinol, if the retinol ester formation is really true for DGAT1, I think it should bind at a very similar pocket, but we are working on that. Yes.
Any other question, comments?
This is Chris Kenny. I have a question. There was a really fascinating talk, and my question comes at a little bit different approach. Some of the substrates for metabolism, they include lipids. If there's the manipulation of the lipid and then inhibition, have you looked or does it change the metabolism of the cell from using, fatty acid beta oxidation to more glycolysis or something like that? Has that been looked at? Or does it even affect it?
Yeah, actually, DGAT1 in mammalian cell, at least, DGAT1 is major, together with DGAT2 as major TG formation enzyme. If you block DGAT1 activities, you can see this DAG will be converted to phospholipids. So this will change the phospholipids metabolism. And that's what we saw in the cell study. And in our mammalian study, which we delete either DGAT1 and DGAT2, they show different phenotypes. For example, in DGAT1, if you knock out DGAT1, the animal appears to be metabolically healthy. They have less body fat and increase the sensitivity to insulin and also glucose. So they show improved metabolism. However, in DGAT2, if you knock out DGAT2, you have severe skin problem. For example, in mouse, the mice died right after birth. So that's what we also see in the knockout animals. Yes.
Okay, thank you.
Hey, Xuewu, this is also Chris, but Chris Sander. It's good to see you again. Really cool looking structures, really nice science you've done. I was just wondering if you think there might be a potential for the tetramer that you weren't able to get a structure for to have some type of cooperativity? If maybe.
Yeah, sure. I don't have time to show that, but it's been long time believed that tetramer is the less active form. I think I still have some time. I can show this a little quick. So, which is here. So this is a cat. Okay, can you see the screen?
Yes.
So this is a tetramer format you viewed from the cytosolic view. You can see this tetramer. This is one dimer, and here's another dimer. Actually, this lateral gate is blocked by the nearby dimer, which could limit the substrate access to the reaction center. So I think that's why people believe the tetramer is the less active format. And I think that's also what's pursuing, because we just don't have enough time to do that. And if you look at the negative spin of the tetramer, they are not bad at all. So
right.
we can work on that for sure. Yes. And also we have enzymeology to show, okay, this is really the less active format. Yes.
Thank you.
This is Philip. Very nice talk. Very nice work. So I think based on the chemistry, the side chains could either be making your carbon a better electrophile, or they could be improving the nucleophilicity of the hydroxyl group. Based on the location with respect to your substrate-bound form, do you think that it looks to me like it's doing the former, so it probably is interacting with carbonyl. Did I interpret the structure correctly? Is that what you're thinking as well?
I think that's what I thought in the beginning. These are very close. Here is next to this carbonyl carbon here, which makes sense that this histidines are attacking the carbonyl carbon here to hydrolyze this cell ester bond. But another essential residue, asparagine, I think is doing something on the hydroxyl group or on the other substrate. Maybe deprotonate the hydroxyl group in that. But in the other structures published in the same issue, ACAT1 to ACAT1 structure, to ACAT1 structure and another DGAT1 structure, they propose actually this histidine is attacking the hydroxyl group on the DAG, which makes sense chemically, right? But because we don't have the other substrate here, they don't have either, which makes me highly suspect their mechanism. But I do think here attacking the carbonyl group makes sense, at least based on our structure. And I think yes.
Did you try doing an aspartic acid substitution for the asparagine at 378?
Yeah, that's also what we plan to do. Yes, but due to this, I mean, you expect to see if by replacing that with aspartic acid, the activity could be, decreased, right?
It could be. Yeah.
Yeah, I think that's what we,
Or it could be a better proton acceptor if that's what's needed, right? It could somehow. It's hard to say. It's hard to predict it. It could go either way.
Yeah, but you know what? To answer such questions is sometimes not trivial, right? And another idea is to add another, substrate. So we actually we found some other acyl-CoA mimics which you don't have the ester bond, but has an ether bond, and that could be a slow substrate. So what we're trying to do now is to add that as a substrate mimics, and on the top of that, we are trying to add another substrate to see how the structure looks like in the active center.
Nice.
Maybe we can capture a different, intermediate state.
Nice.
Okay, any other question, comments? Okay, then the final word goes to Grzegorz.
Sorry, I had to unmute. It's very nice work. I very much like the cholesterol binding. It's very nice to see.
Okay, thank you very much and goodbye to everybody.